Hello everyone, my name is Kaushal Sumaria. I am a PhD student advised by Professor Leo Liu from UMass Amherst. My research is on dielectric performance of parylene-modified PDMS for insulating stretchable electronics. Our work here is motivated by a lot of development in the field of wearable and implantable sensors. The key part here is that the implantable and the wearable sensors both face harsh conditions. For example, sweat and dust, or salt and iron situations. So let's take a look at the biomedical materials commonly used. If you look at the human anatomy, most of the things are concentrated around or less than 10 megapascal. However, the materials used for these sensors are either very stiff in the 1 gigapascal range, or something softer like PDMS, or something even softer like hydrogel. However, the PDMS and parylene are really useful because there are very mature techniques for fabrication of these materials. So let's take a look more closely for these two materials. For parylene, we see that it's rigid while being biocompatible and transparent, really good for imaging, and it can be fabricated with microfabrication. We see some really nice examples of neural probes or retina probes made with parylene here. Parylene is also known to be a good dielectric and used commonly for sealing of PCB circuits or other electronic components. In comparison to parylene, PDMS is more compliant. So apart from being flexible, it can also be stretchable. It also exhibits some really nice properties like biocompatibility and transparency. PDMS is also manufactured by microfabrication, and we see some examples of neural probes or even wearable interface probes here made with PDMS. PDMS is also known to be a good dielectric, however, with the condition that it's thickness dependent. So very thin PDMS cannot be a good dielectric in certain conditions. So if we summarize the material properties of these two important materials used in sensors, we see two stark contrasts, one being the stiffness of the material. Another thing being that if you look at the parylene here, it can resist the diffusion of ions very well, whereas PDMS, being an elastomer, is known to allow diffusion of ions inside, and then that changes some of the material properties that are important for the sensors. So ideally, a material that we would use for a sensor fabrication or for the sealing part, that should be stretchable to be compliant with the human anatomy, as well as provide a good resistance to the ion diffusion. So for that, there are two popular or commonly available modifications that can be done to PDMS using parylene. So we are combining both the materials of interest. One is the simple fabrication of sealing the PDMS with a layer of parylene deposited on top. Another one is a more complicated one where the parylene deposition is done at a higher temperature that expands the pores of PDMS and clogs the PDMS with parylene. So we test these two materials in the electrochemical systems and see how they behave when they are in the ion-rich environment. So our fabrication process starts with a silicon oxide wafer, followed by deposition of chromium and gold layers. For the parylene deposition, we do a 1 micron parylene deposition. For the rest of the three samples, we start with a 10 micron spin-coated PDMS, and then followed by fabrication of room temperature parylene-shielded PDMS or a higher temperature parylene-caulked pc-PDMS. To maintain the uniformity, all three parylene depositions are done in the same session so that we know that the thickness of the parylene deposited is uniform. Yeah, for the testing, we use a 3-electrode setup with a potentiostat. So the dielectric material, so the potentiostat outputs current between the counter electrode and the dielectric material, and the reference electrode measures the voltage. That's how we can calculate the impedance. The bath we are using here is a 1 molar KCl to provide a high ion-rich solution around the dielectric being tested. So the 4 samples are tested, which look like this one after the fabrication. So in order to maintain a fair test, we test all 4 samples in the same bath simultaneously. While measuring, we do a 1-week test, measuring the impedance of each sample every 1 hour, so that we have a continuous curve. We maintain the 1 molar KCl iron-rich solution by sealing the container so that there is no evaporation. And then we test it at 37 degrees to be uniform with the human body temperature. The EIS is done across a large range of frequencies so that we can see its response for different frequencies. So now, before we look at the results, let's see how ideal ion percolation or a diffusion process would look like. So initially, we have the dielectric that is completely free of any ions here. And then once it's subjected to an ion source, the ions will start percolating, and it will divide the dielectric into 2 parts. The one that is completely ion-free, closer to the gold electrode, and the one that is diffused with the ions closer to the solution. Based on this, our model considers these 2 parts, the thickness of which varies over time as the diffusion process progresses. So we simulate this in MATLAB. So if you look at the ideal dielectric or a capacitor, that would look like a dashed line shown here with the phase plot at -90 degrees. Our dielectric that is simulated shows similar properties when it is free of any ions. Once more and more ions are percolated inside the dielectric, we see that the impedance has a shift, and then ultimately it flattens out. Physically, that means that at that point, instead of showing a dielectric property, the dielectric layer is actually showing resistive properties, and the flat line should be a summation of the resistance from the solution plus the resistance of the dielectric material already percolated with ions. So we look at our experiment results. We see that the parylene layer we have demonstrates the behavior closer to a ideal dielectric behavior, whereas the PDMS layer that we have, it captures the ion percolation process similar to what we show in the diffusion model. These 2 are our reference samples so that we can test the layers individually before we test the combined layers. For our parylene-shielded PDMS, we see that the ps-PDMS fabricated at a room temperature process, that one shows a behavior which is closer to the ideal parylene or ideal capacitor. It captures less quantity of ion diffusion, whereas the pc-PDMS shows the condition similar to a completely percolated dielectric layer, where it captures a lot of ions inside it. We also plot the evolution of impedance over time for 3 different frequencies. And if you look at the trend, consistently the parylene layer performs better than the 3 other layers. The PDMS and the higher temperature pc-PDMS, it deteriorates much more compared to the other samples here. So let's review what we see until now. We see the ideal diffusion process and the samples that we tested. So what we see here for the sealing of sensor devices, there is no ideal material that can show stretchability as well as sealing properties. But with clever engineering, we can find a middle ground where the ps-PDMS is able to provide better sealing compared to the pristine PDMS. And then we can also alter the stretchability of it by some engineering methods. So we think that ps-PDMS is a good candidate for the sealing of sensor devices. So in conclusion, we have a testing setup that closely represents how the sensor would face the situations when it is implanted or interfacing with the human anatomy. Then we provide a diffusion model to see the insights in the process. And lastly, we test 4 different samples for their behavior under ion-rich environment. And then lastly, I would like to thank the funding agencies that facilitated this work. and I would like to take any questions.